to all of our mom geeks out there. Thank you all for joining us tonight for our May virtual meeting of the Wake County Beekeepers Association. Hopefully, anyone who started with a nuke or a package over the past couple months has had the good fortune to see them flourish and not swarm. Our swarm captains have been keeping busy, making phone calls and sending out some folks to rescue those renegade bees. If we have any of those zone captains on the call tonight, if you can just possibly type in numbers of how many calls and how many swarms you've actually sent folks after, that would be helpful. I apologize, I didn't think to ask Peter for that number earlier this week. Any of those of you watching, your hives and your landing boards or your entrances will probably agree that there's a lot in bloom out there, and my allergies would certainly agree with that as well. Susan Benton mentioned to me just a few minutes before the meeting that she's pulling some frames tonight and going to start extracting some honey this week. So anyone else have the good fortune to be pulling frames already? Let's see a show of hands. That would be interesting to see. I've seen a lot of people talking on Facebook that they're sort of amazed at how quickly frames are filling up this year. So good luck to all of you with your honey flows. Before we move on to tonight's presenter, just quick show of hands to anyone that might be joining us for the first time tonight. And looks like we have a few people, so welcome. Hope to see you back again for many more meetings, whether they be in person or virtual. So without further ado, I'm going to give you a quick overview of tonight's agenda. And we're going to move to our presenter for this evening. I'm going to turn it over to our program director, Jeremy Hayes, to present our presenter. But before I do, I'd just like to personally thank Dr. Esmaeil Amiri for joining us tonight, and a reminder that we'll be monitoring the chat. So if you have any questions throughout his presentation, please post them there. And as always, if you could remain muted until after the presentation is complete. And we may open it up to individuals asking questions, or we may just have one of our monitors do that for us. So I'm going to stop sharing now so I can turn it over to Jeremy to present Dr. Amiri.
Hey, everybody. Hope everybody's having a good week. Dr. Amiri, go ahead and you can take control of the screen. I don't need the screen. Dr. Amiri is going to introduce himself, but I'm just going to say a couple words from bio he sent me. He's a migratory professional beekeeper from Iran with undergraduate studies in several European countries, including Germany, Sweden, and the Netherlands. He's got his PhD from Denmark, and he works with Dr. David Tarpy over queen virus infection and honeybee egg characteristics. So looking over his credentials here, he seems to know a lot about virology, words I can't even pronounce, virus host interaction, virus pathway. So this is the guy to talk to if you've got questions about honeybee diseases.
Yeah.
Dr. Amiri, go ahead.
Thank you so much. And hello, everybody. And I'm very happy to be with you tonight. But first of all, I want just to apologize you because I'm not a native and I do have a strong accent. If I pronounce something completely wrong, just please forgive me. But I try to do my best. And I was asked to talk because I can't talk about the honeybee viruses and specifically viruses on honeybee queen, but because we did a study about the honeybee egg size, And we think that it's a fascinating phenotype in honeybees that can be in future be used for breeding or something like that. I decided just to present this topic to you. I don't exactly know how much it will help you as a hobbyist or professional beekeepers, but hopefully that you will enjoy it. And as Jeremy said, yes, I'm a native of Iran, and then I was a beekeeper there. I did a beekeeping like a professional and migratory beekeeper for some years. And then I got a chance to move to Europe. And due to my my personal business in Iran, being a beekeeper, when I moved to Germany, and then first of all, I moved to the Netherlands, and then I moved to Germany, Sweden, and then Denmark. And every place and every country that I have been there, first of all, I wanted to learn about the beekeeping and beekeeping culture. And that's interesting. And I hopefully one day I will make a presentation about different culture of beekeeping in different countries. Because it's not what I understood, and most of the people think that beekeeping is, okay, you have a box and you have a bees in there and you do the same thing, and every country is doing people in different countries are doing the same thing. But when you deep down to the culture, you see, no, things are completely different. When it comes to beekeeping culture, things are different. Every country has its own culture for keeping bees. So I'm hoping for that as well, to one day sit down and make a presentation, present it somewhere that everybody will enjoy it. But tonight I will start with the egg size. And we know that how much egg is important for the colony. Egg is one fascinating stage of the developmental stage of honeybees, because it's a connection between queen and the next generation. And one egg can, with the plasticity of one egg, can turn it to be either queen or worker bees. If it turns to be queen, it will develop in 16 days with a special food. Everybody knows about royal jelly. But if it turns to be worker bees, it goes with 21st days and then receive different food. So you see one egg, the fascinating characteristic of eggs that can be either queen or worker bee. So it is one important aspect of developmental stage. The other interesting thing is that in honeybee colony and specifically on social insect, the queen is just responsible to produce the egg, and she doesn't have any responsibility to take care of it. And it's mostly and it's only more worker bees to take care of the egg and rear them and develop them. So you see how these things, and then the egg can be connection between queen and worker bees. So it's very important. Egg has many characteristics as well. You know, in part of my studies, I study honeybee viruses and how viruses transmit from one generation to another generation, from one individual to another individual. Many times we find a queen being infected by different viruses, and it's interesting that the viruses can transmit through the egg, going from one generation to another generation. And the good thing in here, even though it's viruses by themselves are bad, but the good thing is when the queen get infected by viruses, for some reason, queen will become like a benign. It's not turning to be acute. It's turning to be benign and not hurting the queen by itself for a long time. And queen can resist the virus infection in many cases, not always, but in many cases. But and it's because the virus, if the virus kills the queen, then it's the end of the life for the virus. But virus stays as a benign and then transmit to the next generation through the eggs. And while the viruses are infecting the queen, that is another thing that is under studied right now with different researchers, is that when the queen is infected by viruses, still she is challenging with viruses, still she is fighting with viruses. And while she is fighting with virus, she produces eggs. And because she is challenging with virus, she is producing so many immune genes and immune proteins. So when she produces eggs, some of these proteins, she just put them in the egg and then transfer them to the next generation. So there is different ideas that queen can immune her progeny by doing that putting some immune proteins and immune genes to the eggs and make the next generation a little bit more immune. Because as you know, eggs should be by themselves for 3 days until they hatch. So she needs to put enough food and enough components to the egg for the egg to survive for the first 3 days of life afterwards. So that is two ideas that is around. But going back again to the egg and egg characteristics, you know that queens most of the times needs to just put 1 egg in each cell, but many times they put 2 eggs, and many other times they put more than 2 eggs. They are not workers that laid eggs here. I know them because I had a queen, and then for some reason she did not like to put eggs in other cells. She did like to put eggs only 1 cell. I don't know for what reason, but most of the cases that I noticed having 2 eggs in one cell. And that is interesting because we, as a beekeepers, we think that at the beginning of when the queen is young and just started laying eggs, she is kind of confused and she produces 2 eggs at once and lays 2 eggs in 1 cell. But me working with honeybees, many times it's not correct, and different reasons can cause that. Sometimes the infection in the ovaries can cause that. Not the high level of infection, but some level of infection. The other time is sometimes we have the queens in the small colonies. They come as a nuke, because if you have seen it, queen producers have these queens in the small colonies in the mating nukes, and then after they are mated and they lay some eggs, they catch them, have them in the cage, and send them by packages. But then because they were like kind of got used to that, the small nukes, when you introduce them to the big colonies, they become kind of confused and lay 2 eggs or 3 eggs in 1 cell. I have seen it because we moved, I will talk about it later. We moved the queens between big and small colonies several times, and we wanted to see what is going on there. And we saw every time that we move queens from small colonies to big colonies, for 1 or 2 days, they are kind of not knowing what they do, and we noticed that many cells having 2 eggs in one 2 eggs in 1 cell. So queens can have eggs in different shape and different size. It's not, if you think sometimes back when I was in Denmark, my supervisor I did talk to the beekeeper, and he told me, queen producer, he told me that always he is grafting from the queens that lay big eggs. And it's because big eggs tend to be big larvae when they hatch. And then when I talked to my supervisor, he said, no, all the eggs are similar. They are just the small eggs. They are not different. So then when I came here, we got the project, and then I started just to measure the egg size and look at the eggs under the microscope. And as you see, different shape, different size, and it got like more fascinating afterward. And that's why we started, look here, 2 eggs, 1 is big and 1 is small. How different they are, you see. And that's why I got more interested by looking at the eggs under the microscope. I was interested to understand what is the cause of these differences between eggs from one queen to another queen or from one colony to another colony. And that's why I came with two ideas. Either it can be genetics or it can be environment. So we wanted just to test all of these and understand it completely what is going on. And that's why we had three hypotheses about the effect of genotype on the egg size, effect of colony size, because if you have a colony with let's say nuke, there are only 500 or less than 500 bees versus colonies that has 50,000 bees, or if the nutrition is up and down, what will happen to that? And that's why we started just to analyze and study this phenotype. First of all, we bought queens from different resources in the first year, and we bought queens from 6 different different genetic background, and brought them all those queens. They shipped to our department, and we introduced them to the small nukes, as you see here, and had them all in one apiary. We fed them. We did everything at the same for all of these colonies. And after the colonies started growing, and the queens established themselves, we started to measure the egg size. What we did, we had the empty combs. We caged the queen in this empty comb for one night, 15 hours, almost 15 hours. And day after, we released the queen, cleaned the frame from the worker bees, and took the frame to the lab. And then individually, we took a picture, as you see here, from each individual egg for 20 to 24 eggs. And as you see, we used the software called ImageJ, and then we clean up around the eggs, and you see in the bottom picture, and the software gives us the area about this egg. So after that, we analyzed the data, and we understood that they are completely different. For example, we had queens from California that had the biggest eggs, and on the opposite side, we had the Hawaiian queens from Hawaii that they had the smallest eggs, and then hygienic, USDA Paul, and Russian in different sizes as well. But these two were like kind of interesting, why one set of queens are producing big eggs and the other set of queens are producing a smaller egg. So that was kind of getting to know that, okay, the genetic of the queens has big impact on the egg size. So then we wanted to test the environment. First of all, what environment comes to mind is just the different size of the queen, the colonies, because as you see, and I explained that we had queens in the nuke that there are only 500 bees in there, and then we had mid-sized colonies around 3,000 to 5,000 bees, or big colonies around let's say 30,000 or 40,000 bees. And our thought was, because the queen in the big colonies are more in contact with worker bees, or there are more worker bees there, so these worker bees are feeding rigorously the queens, and queens will have more food, they produce bigger eggs. And then on the other hand, when the queen is in the small colonies, because there are only few eggs, only few workers, the queen will receive less food and then produce smaller eggs. But in reality, it was not correct. After we analyzed the egg size as the method I explained, we understood that queens in the small colonies are producing bigger eggs, and queens in the large colonies are producing smaller eggs. And interestingly, queens in the middle were produced the egg size as in the middle of those two extremes. So that things become more interesting. And that's why we wanted to test it. Is it correct or it's not correct? And that's why we got another experiment. We got another experiment and said, okay, let's just try it another way. We got queens in the big colonies and we collected samples for 3 different weeks. And then after 3 weeks, we moved the queens to the smaller colonies. And then again, collected eggs for 3 weeks. As you see, in general, when you see the left-hand side with that, all the eggs together from big and small colonies, it's completely true. Queens in the larger colonies produced smaller eggs, and queens in the small colonies produced big eggs. When we come to the individual level, every queen, we had 5 queens. I mean, we had 6 queens, but 1 queen died after when you move the queen from big colony to a small colony or vice versa, you are not always successful. So we lost 1 colony, but in general, the same trend is correct for all of the queens. That is good. But the other thing that we understood is that there is a differences between or among the queens in each group. For example, for 3 first weeks, we collected eggs. You see, all these colonies, all these queens produce different size of the eggs. And then week to week, it's a little bit different. And it's depend on the environment, what type of food they bring, how rigorous they can bring food or nectar or pollen or so on and so on. Even that, the other interesting was one of the queen that we had, so she changed the egg size from a small to big. The first week was fine, but the second week, she just reduced the egg size. For what reason, we have no idea. And then she tried just to turn back, but she didn't have more time for more week to find out if she's completely back or not. But this is how it is. And the other interesting thing is here that about this extra one, it's the individual line that you see here. This queen was producing big eggs when she was in the big colonies. When she moved to the small colonies, even she produced more, I mean, the biggest size of the egg. So what is going on, we don't know. Maybe part of this explanation, I can talk about it in the end. Then we said, okay, still it's not fine. We need to do more. We produced 12 sister queens from one single mother. And then we introduced them, or at least when they were in the queen cells, we introduced those queen cells in each mid-sized colonies or 5 framish nukes. And we did let the queens to emerge in the same colonies, in their colonies with the same number of worker bees. And then after they started laying eggs for 2 or 3 weeks, we divided them. We divide half of them to the small size colonies and the other half to the big size colonies. And we wanted to see, okay, now they are sister queens. We want to see what is going on. After collecting eggs for 4 weeks, 4 different weeks, we understood the same pattern is still true. Those queens in the big colonies that you see in the bottom of the graph, in the big colonies, they produced a smaller size eggs. And those in the small colonies, they produced big size eggs. So that was kind of interesting. And that's why we try to go deeper to understand a little bit more. Is there food playing a role, or what is exactly going on with the small and big colonies? Because when you think about small and big colonies, of course, when the queen is in the big colonies, she is more covered with many bees, and she receives more food, And if there is a stress from outside of the colony, she is the last individual to receive that stress. But then if she is in the small colonies, that is completely different because she will notice the stress very quickly. And that's why this stress, we decided to have it as a pollen restriction or overfit. We had, again, 12 sister queens. We had 6 of them to emerge in the mid-sized colonies. It's in 6 colonies, the same size of the bees, and the other 6 in the other 6 nukes, or 5 Flemish nukes. With one group that we started to feed them every other day with sugar syrup. With the other group, with the pollen restriction, we had a queen excluder or pollen, what it's called. We didn't, let's say like that, we didn't let them to take the pollen inside. And because worker bees eat the pollen, the idea was that worker bees eat the pollen and feed the royal jelly to the queen. So we said, okay, if there is less pollen, these worker bees cannot produce that much royal jelly, and queen will be impacted by that. And then after 4 weeks, after 4 different weeks, as you see, the first week, August 28th, even the queens in the restricted eggs, even though here was not a restriction, but the egg size for this group were a little bit higher. But when the time passed, the queens that were receiving overfed, they did not change the egg size, or the egg size remained as similar. But when there was a restriction for the pollen, the egg size started to become bigger and bigger. So that is what we understood. We understood that the queen is looking to the colony and understanding what is going on, how much food is coming to the colony, how much nectar is coming, how much pollen is coming, and all these things. And then she will, because one idea is that when she sees bad situation, or it's almost with all of the animals, even with animals and other insects, where the situation or environment is not good, they put more things to the egg and make the next generation kind of safe. But when the environment is safe and they see, okay, enough food is around, they put less in the eggs. And they know that the environment is good enough for the queen, for the next generation to survive, let's say that. Okay, so then that was the idea. Then we decided to say, okay, now it's good. We understood that the egg size is changing by environment, but what is the effect of this egg size on the survivability of these worker bees? That's why we cage the queens again in the big and small colonies, and let them produce egg for one night, because we wanted to have similar egg age for all these queens. And then after all the queens, after 12 hours or 15, let's say 15 hours, it was 15 hours. After 15 hours, we release the queens and we count how many eggs every queen laid. And for 3 days, we did let the eggs to stay in their own colonies to hatch, become larvae. And to be sure that if we move these combs to one common colony as a nurse colony, all of them will survive there. Worker bees from the nurse colony will not remove them. So all of these eggs turn to be larvae, and then we move these larvae, the combs that were containing larvae, hatched larvae, move them to the common environment, one big colony, to be all in the same environment. And then for every stage, we went back and count, okay, how many eggs at the beginning we had, how many eggs hatched, how many eggs went to the next level, become larvae, become pupae, become adult. As time passed, as you see here, as time passed, we understood that the larger eggs survive better than smaller eggs. And it's interesting because at such that environment, we decided to have them at the worst situation, because if you put them in the right and good situation, all of these eggs with all of these larvae become worker bees, or you don't see the real effect of it. But if you put them in the worst situation, it was end of the year in August or sometimes, and then not much of nectar and pollen is coming. The colony did not have enough worker bees to take care of all of these. And we understood that larger eggs survive better than smaller eggs. So then we decided just to continue and make it a little bit more complicated. So that's why last year, again, we repeat the same experiment with a little bit more complicated situation. So we produced a bunch of queens, introduced them to these 5 frame-ish colonies, because we want them to be in the middle for the experiment. And then half of the queens, we moved them to smaller colonies, and the other half, we moved them to the big colonies. In every step, we were collecting eggs and we were measuring the egg size. And then after 2 weeks, we moved the queens from the small colonies to big colonies, and we moved the queens from big colonies to small colonies. And every time we measured the eggs. After that, I mean, we did this project with Dr. Han Bin. He came from China. He had kind of a scholarship from Chinese government. He came here and we worked together. Nice guy. So after that, we analyzed the data. And as you see, interestingly, the queens, again, the same result did happen. The queens in the mid-size that you see, they, at the beginning of the egg laying, the egg size was not fixed there because they got mated, they came back and started laying eggs. We collected eggs and measured the egg size. Still, the egg size was not fixed. And then week after, again, we measured the egg size, and it became more fixed of real size that one queen will lay eggs. Then after that, we divided the queens. Half of them went to the small colonies and half of them went to the big colonies, so large colonies. And as you see, those queens that were to the large colonies, they laid a smaller size eggs. And I said we collected 2 times eggs. And those queens that were we moved them to the small colonies, they started laying big eggs. And then 2 weeks after we collected eggs, we moved the colonies to queens from the smaller eggs to big colonies. As you see how different the size of the eggs change, and the other way around, we moved the queens from the large colonies to small colonies, and the size changed completely. So then that was good. I mean, we could confirm that what result we got with the previous experiment. Still, we are able to repeat the same result. So this is one strong proof about that. We tried to analyze them. Han Bin got the samples to China because he had all the machines and analyzed the proteins of these queens. And as you see, at the beginning, we had group A and group B that they produce big eggs and smaller eggs. And as you see, completely different protein profile they have, okay? But then after we moved them, for example, this group B to the small colonies, they started laying eggs. And you see this, they become group B, become group C. Of course, we lost a few queens because we moved them from big colonies to small colonies. We lost a few queens, and that's why. But when we move the queens from big colonies to small colonies, the protein profile of the eggs changed. And then the same the other way. When we moved group A, that were in the small colonies to large colonies, you see group D, for example. It did change, but not completely. So we are trying to analyze this. Even we analyze the ovaries of these queens. What is going on with the ovary proteins? And the same story did repeat. I don't have the graph here, but the same story did happen with the ovaries too. When we move the queens from one from the big colonies to the small colonies or vice versa, the protein profile of the ovaries and eggs change. So one will ask, Esmaeil, what is the difference? What is the use of all these things that you have done? The thing that we are looking at is and then we continue the experiment. The experiment, we measure the egg size, we measure the 2 days old larvae, 3 days, 4 days, and 5 days. And you see after 5 days, the larvae, the cell capped, and we don't have any access to that. But the thing was, we wanted to know the growth rate of these eggs. If they are a small size, what is the growth rate if they up to day 5 or 6 after hatching? If they are a small egg size, what is the growth rate? Because part of this one, how much the queen put in the egg, that is what the queen put. But then we wanted to know what is the effect of that when we have all these eggs in one colony. Is there any role that workers are playing to compensate this small and big or not? The analyzer, I don't have any data to show you this tonight, but the analyzer is showing, yes, there is some differences with the growth rate as well. We would like to just continue the experiment and next time have it as find out the survivability or the activity of the queens. We have RFID system. The RFID system is like a tag that we glue to the bees. And as you see in the other picture, this is not from us. I just borrowed from a friend. But the system, we have the system here in the university. And it's like that's a clothes that you go to the shop and buy from there. If you pass the door without paying it, it starts beeping, and it's exactly the same thing. So every time bee is passing this reader, it just collect the data. So we will introduce it. We will rear these bees from small and big eggs, and then after they emerge, we will have the tag in the back of them and collect data to understand how fast they start working in the colony, outside of the colony, how many times they go in and out, what they do. I mean, this kind of information. And the most important things that we would like to understand is the longevity, because it assumed that these workers from the big eggs, they survive 1 or 2 days more than other workers, the workers from the small eggs. And when you think about the whole colony, all these worker bees survive 2 days more than the smaller size eggs. We see how fast the colony can grow and how long one queen can stay alive in a colony. So this is our next step to work with. With that, I would like just to conclude what I said, that our understanding is significant differences between size of the eggs from different stocks, different genetic background has effect on the egg size. Eggs from the queens in big colonies are smaller than queens in small colonies. So when you think about the small colonies, they produce big eggs. When you think about the big colonies, they produce smaller eggs. And one reason is because the colonies in big colonies, they bring more food. That is correct. But at the same time, they need queen to lay rigorously to keep the number of colony, the number of bees growing. And that is why queen doesn't spend so much on so much energy and put so much protein and genes inside each egg. So she just do the minimal work for her and lay eggs. Results suggest that egg size is actively controlled by the queen. Queen is just actively looking how much food it comes to the colony, how much nectar, how much pollen, what is going all in the colony. Even though we think that queen is not short in food in the colony, like if you think about like solitary bees are normally, they look for food by themselves and then they lay eggs. If there is less food, they lay less eggs or bigger eggs in this regard. But in general, they lay few number of eggs. But when we think about the queen in the colony, in a honeybee colony, we think that because worker bees are feeding her, she is not short in food, but it's not correct in some way. That she is looking around the colony and understanding how much nectar and pollen is coming to the colony, and based on that, she controlled the size of the eggs. And then the last things that I can say, offspring of queens that produced large eggs survived more than offspring of queens that produce smaller eggs. So that is the finding of our experiment. So we are molecularly analyzing the eggs to understand what proteins are changing, what is the role of each protein for survivability, for longevity, and all those. We are hoping that by the end we get like a comprehensive data with a solid result that say, okay, if in future queen producer or some other people that would like to do these things, if they want to produce or select for the trait, and if we understand that this trait as its egg size can help for the survival, can help for let's say resistant to some diseases, we hope that our result will help that we come to the end and we have a suggestion for them as well to select queens that produce big size eggs for the health of honeybee colony in general. So by that, I would like to thank my mentors, Dr. Olav Rueppell, David Tarpy, and Michelle Strand, and a student that helped me, and all the funding agencies that funded my project for these several years. Thank you so much. I'm happy to answer your questions if you have any.
Great, thank you, Dr. Amiri. That was very interesting. I'm not sure, Jeremy, are you fielding the questions tonight, or shall I? Since we're not getting a immediate response from Jeremy, I'm going to just go ahead. If you don't mind, Dr. Amiri, if you can stop sharing your screen.
Yeah.
And we'll jump into, so I have some questions from the beginning of your presentation. We have a question from Tom Krupa. He's asking, how does the queen attach the egg to the bottom of the egg cell?
Oh, that is interesting. Recently, there has been somebody from Germany. He recorded the queen that lays eggs in the cells. I recommend you to see it. It just got viral everywhere, here and there, and it's interesting that she just put her butt in there and find the end of the cell and put the eggs in there. That is interesting. I recommend you to find that. It's 1 month, or it's released like in a month or so, if you can. And the article, because he filmed the colony in some way to find out, okay, when the queen lays egg, she just lays egg and leave. And then afterward, worker bees come and do something. And it's interesting because our understanding was that these eggs only need a temperature and nothing more than that for the first 3 days after egg laying. But it seems that it's not true. Worker bees come and then somehow untaining these eggs or doing something, and nobody knows what is that. That is completely new. And it's become interesting because there is a professor in Illinois University, and they developed a machine that it become like a worker bee. And at the same time, they have all these eggs, and they want just because they want people want to feed when the colony, let's go from the other way. When you have the colony, you have eggs and larvae in the colony. So you see many pathogens, microbe, and everything is around. And when worker bees are feeding these eggs or larvae, those pathogens will be involved in this interaction. So in the Illinois professor, he is interested to find out just have the sterilized food to the larvae and worker bees until until they emerge, and then understand when they get infected, how much they get infected. If he introduces different stressors, not only disease agents, but then. After this movie released, he started thinking maybe the way that he's thinking to work is not correct, because worker bees during those 3 days are doing something to the eggs. It's not just have them, queen lays eggs, and then nobody cares about them until they hatch, but it's completely different things. So this is what I can say. My recommendation for you is just the movie got everywhere. I'm sure you can easily find it. You can look at it and understand this situation.
Okay, if anyone happens to find that in the next couple days, go ahead and if you can post that or the link to it, that would be appreciated. Another comment from Chris Hagwood. He says maybe waiting for open cells in smaller colonies results in larger eggs. Maybe test by caging them.
With caging is not a big deal, because even for 15 days, we reduced the caging to 6 hours and still we got the same result. I mean, the caging is not the big deal. And even having queens in the small colonies, we had them, and one idea that we had, because we have box and we have a colony, that is 2 different things, which sometimes we mix them, okay? Then we decided to provide more open cells in the colony to see if there is availability of open cells. Still, she lays more eggs and then just leave them until either worker bees take care of them or not. But that was not true. Queens lay eggs as to the number of workers that they have. This is what I can say. If they have less workers, they lay less eggs but bigger eggs. If they have more workers, they lay more eggs but smaller eggs.
Okay.
Yeah.
A comment to follow that, to follow Chris's question was from Jim Bly, and he was saying, or maybe laying fewer eggs results in bigger, laying fewer eggs results in bigger eggs.
Would you repeat it again, please?
Sure. This was just a follow-up comment that Jim commented to Chris's, the previous question, and that was, or maybe by laying fewer eggs, it results in bigger eggs.
Possibly, but our result did not prove that.
Okay.
Yeah.
Another question coming from Jim is, are eggs laid in a queen cell larger than those in worker cells?
We did not do that, but a group in China, they collected those eggs. So they collected, they have a system, I think they use it in China, that instead of this genter system that we use here, we have this genter system, and then I think it is genter, and we have the half size cage shape, and we have the queen cage in there, and queen after a few days start laying eggs in there, and then it's horizontal. Then when they turn to be larvae, we take them out and turn them to be vertical to produce queens. But in China, they have different system that they can use it, and they have it as a vertical, as a vertical from very beginning, and queens lay eggs in those. And that's why they collected eggs from those queen cells versus the eggs in the worker cells, and understood that that is correct. That is really, really correct, that eggs in the queen cells are bigger than eggs in the worker cells. Even they went ahead and then analyzed them, and they found the same thing. They found that when queens originally lays egg in the queen cells, genetically and by protein things, they are different from the eggs in the worker cells. So that is correct. Yeah, I agree with you. And there is scientific evidence behind that.
Okay, thank you. And another question here from Russ Bullock. Is there a difference in the number of eggs produced per day? For example, a larger colony has more eggs and a smaller colony has fewer eggs.
Of course, yes, yes.
Okay. Okay, and then Robin Dreschler is asking, how did protein profile change, either by number or variety?
Because I did not do the experiment, I'm not in details. What I showed you, this is the big picture I got from the collaborator from China.
Okay.
Yeah, I cannot go deeper than that. And hopefully you did not record, or I see you are recording. Hopefully you will not release it on YouTube, because those few slides in the end, I mean, these I wanted just to present it here. We don't want to, because the data are not published yet. I would be appreciated if you just have it for yourself.
Our stuff stays within our club, so no
that is,
to
yeah.
YouTube.
Yeah, that is fine. Yeah.
Okay, and then this the last one I see is actually a question from Jen Fawcett and myself. So do larger egg size equate to larger bees, and is there a benefit to having larger bees?
That is what we are trying to understand it. That is what we are trying to understand. And that's why we collected, because we wanted to know when these size, either these size, when we have a smaller eggs and big eggs, and worker bees are feeding them, either they will stay as a different size until they cap, the cell caps, and how they produce when they become like a little bit bigger or smaller, what is that? And that's why we collected bunch of different size from different life stages to understand it. We are analyzing, and what we want to understand, first of all, we want to understand it will be like the different size for all these developmental stages, or at some point the larvae size becomes similar between those two different groups, and we are analyzing it. I mean, we would be able to have the results sometimes this year or early next year.
Okay,
Yeah.
excellent. Thank you. I think I covered all the questions. I think I grabbed everyone's questions. If I missed your question or you have additional questions that have popped up, you can go ahead and unmute yourself and ask Dr. Amiri yourself if there's any questions. Anyone else?
I was curious, with the changes in the protein profile, do you notice any changes in phenotype or behavior, or what's your theory on what is increasing the survivability of the larger eggs? Does it have to do with the bee behavior, or is it just the eggs themselves?
We don't have the data for that, and that's why we are using that. We are going to use that RFID system to understand it, how fast they start working outside of the colony, how many times they go out, how long they live in the colony, the longevity, and all those things. But our first idea was that bigger eggs will have higher vitellogenin. And vitellogenin is a protein that is in the eggs, but and it's like kind of immunity or food, or it has different function in the egg. But seems it's not true from the result that I got from the colleague. It's not only vitellogenin. There are other proteins that become involved to it. I have, let me just share something different with you if I find the things, because you seems to be more interested in that. I don't know if you have time or not, or just I can do that. I can do that. I can just talk about these things and then show you how things is different. May I just share my screen again?
Yeah, sure.
Yeah, let me just share my screen this time. Even though it's a little bit different graph, but you will see it interesting. This is what we got from the ovaries, and then you see these small colonies, the queens in the small colonies, and those 3 queens in the big colonies. When we analyze the protein, you can easily notice the protein profile in the ovaries are different. Okay, so this is what I wanted to show. Here is the protein things, and then when it comes, you see proteins mostly become in different categories when we analyze the eggs. Some structural, that is good development, we know it. This mRNA binding are something like very interesting. These protein binding are interesting. And these 4 groups, innate immunity, okay, these are important as well. So how much these big and small eggs, the proteins in there contribute to the immunity, because one egg doesn't have a way to being immune against different diseases or pathogens and so on and so on. So it becomes more interesting here. And this oxidative oxidoreductase activities. These 4 things. The other 2 are part of it. It's not like kind of surprise this developmental and the other things, but these 4 smaller ones, they are kind of interesting for us, and that's why the Chinese collaborator, he is interested to go ahead and do more analysis this year. He went back to China, but the system that we used here is exactly, he's going to copy it over there and understand it, what proteins are changing exactly, and what protein, what is regulating this size, what proteins or pathways or genes are regulating this X size change. What is exactly going on that one queen can control the size and say, okay, now it's a small and then and the big one. So he's trying to get down to that level to understand it. Yeah.
Great. Well, it's all very interesting, and I think that probably we could talk for more another
Yeah,
hour or
exactly.
so. I know that you had asked earlier if once you were done with your presentation, if you could sign off. So I think unless anyone else has any follow-up questions, and if you do, you have about 5 seconds to chime in. Anyone else have any additional questions for Dr. Amiri? Okay, looks like,
One quick one.
go ahead, Johnny.
Did you get any difference between when the egg was laid to when it came out as an adult, time-wise?
No, we did not. We did not do that. But one thing that I can add here, because it came from another experiment, and I would like just to say it because I experienced it by myself. So if any of you are going to produce any queens, be sure that the colony that you are producing queens, they are healthy and they are clean of varroa mite, because that my life is completely different from yours. Okay, I am looking for unhealthy colonies because I want to understand the diseases. I want to understand, but you want a colony to be healthy. So I grafted from one mother colony to 2 colonies, one healthy and one unhealthy. Okay, so one, I introduced mites, diseases, and all those things for sometimes. And the other one I treated, and then I wanted to have healthy and unhealthy colony. And those queens, those larvae that I grafted to the unhealthy colony, of course, there was few queens emerged from that. But my understanding, looking at every the other day, that even the queens did not receive enough royal jelly, and one day sooner they were capped. Okay, so it means that when there is not enough royal jelly inside, or the queens are producing less royal jelly, the queen larvae passed the process very quickly to be capped, and then afterward, when they emerge as a smaller queens. So this is my personal experiment, and I'm working on that project too. So that is, but with this project, I don't have any idea, because as I said, this is like completely new. We don't have so much information, or we don't have information. And for most of your questions from after egg or after larvae, I can say I don't have any result, and I don't have any answer. This is one we found, and I was interested to share it with you.
Well, we look forward to those results or your next study. So keep us updated. We appreciate it.
Sure.
So we're going to switch over to our business portion of the meeting. So if you could give Dr. Amiri a hand, and we thank you for your great presentation this evening. You're welcome to
Thank you so much for inviting me.
You're welcome to hang in there with us, but if you need to go, we understand. Take care.
Sure. Yep. Thank you so much. Have a great time.
Thank you.
Yep. Bye-bye.
Okay, I'm going to go ahead and share my screen. Give me 1 second here. Okay, going to move on to tonight's treasurer's report. Our current balance, numbers coming in from Susan Benton, our treasurer. Current balance is $19,908.61. Bank balance is $19,664.61. We have a PayPal balance for membership renewals of $164. $25 check still to be deposited. Petty cash is on hand is $80, and expenses still to be paid $375 to NC State Beekeepers Association for some memberships that have come in. Speaking of membership, our membership report from Gordon Goking is Our current totals as of yesterday, we have 341 members, 149 new members, 175 renewing, and 17 are 70 plus or lifetime members.
Hey, Kim, I'm not sure if you're trying to share your screen right now, but we're not seeing it if you are.
Oh, okay, give me 1 second. Give me 1 second. Are you seeing it now?
Not yet, no.
There
we go.
Okay, sorry about that. Okay, gonna just go back. So those are just for those of you who are visual people. That's the numbers that we had submitted. Okay, so the next slides, I'm just going to read through these, and my suggestion to you guys, because I know there's some links on here that might be very helpful. I would suggest doing some screenshots of any of these that are of interest to you. I have 3 slides for our old new business announcements and upcoming events. So from the state organization that went out to all chapters regarding our master beekeeping program, they are offering written tests for journeyman master levels on June 26th of this year in Hickory. Pre-registration is going to be mandatory on those, and all approved registrants will be required to sign a waiver of liability to participate. You're going to follow CDC guidelines, limiting it to 30 beekeepers. Approved face coverings are required and social distancing mandated. To start the registration process, there's an email address there, and that registration is going to close on June 11th. So if that's something that is of interest to you, please make sure that you follow up by June 11th. And additionally, Susan Benton reached out to Doug Galloway and requested that we try to get a testing closer to Raleigh, and still waiting to hear back on that. So if if you're able to make it out to Hickory, fine. There might be another opportunity. We can't guarantee when that would be, but as soon as we know, we'll get that posted. Our next meeting is June 8th, and that's going to be another virtual meeting. And as mentioned last month, that's going to be dependent upon our upcoming poll events, whether when and if we'll move to in-person meetings and events for the remainder of 2021. We're limited in different facilities that at this point will be able to house the number of people that we have. Sometimes we have as few as 30 people. Sometimes we have as many as 100 people show up at our in-person events. So we have to be ready to accommodate any of those sizes. Again, we have some hats and t-shirts for sale. If you're interested, you'll need to get a hold of Susan Benton. Dr. Tarpy and the NC State Apiculture are continuing their beekeeping webinars. The next one is coming up on Wednesday, May 26th. The information is there. It's planting a pollinator garden with bee plants. And also, if you're not checking the Wolfpack Waggle newsletter, you might want to check that out. There's some interesting information that you can get through their newsletter. Some of you might have already participated this in past years or already sent your mite count in, but there's just a week left in the Pollinator Partnership Mite-a-thon, and their objective is to raise awareness about honeybee colony varroa infestations in North Carolina. So if that's something that you want to participate in, the website, the link is there. Also this month from Bee City USA and Bee Campus USA, there's the opportunity to download some black and white, which your kids can color, or some color prints for No Mow May, signs that you can post in your yard if your HOA allows it. This is a month-long initiative to help support early season pollinators by allowing your yard to grow for the month of May. And it's just another way to educate your neighbors and the community on different ways to keep feeding your bees. As mentioned last meeting, we're looking forward to being back at the state fair again this year. The dates are October 14th through the 24th, and both we hope to have a booth with our county, and the NC State is hoping to have a honey booth that they can sell honey again this year. So those you probably start seeing different emails coming from the state organization, as well as we get nearer. Usually July is our deadline for submitting things. So in the next month or so, we'll start asking people if they're going to participate. And lastly, don't forget the great resource you have with your current and archive issues of Bee Buzz. There's a lot of information that you can get out of the magazine, and it's basically from area beekeepers all around North Carolina, and they're always looking for your unique photos to use, as well as new writers. So if that's something you're interested in, you can look for the details in one of their issues. And you can download it, or I know that some folks prefer a hard copy version, and that's something that's included with your annual membership. Okay, so trying to get through everything quickly so we can move on to Dana. Just wanted to let everyone know that the Hands on the Hive out at the Oakview Park, we've had great success since April 11th. We've welcomed over 52 beekeepers to the hives, and 25 members have opted to take their practical test, and they've passed. So congratulations to those folks who are one step closer to becoming a certified beekeeper for North Carolina. And here are the dates. I know that there's a current Sign Up Genius out there for the upcoming May 16th. And I spoke to Ben Crowley, and going forward, it looks like we'll open it up to our normal third Sunday of the month from May through September. The address is there. The dates are June 20th, July 18th, August 15th, and September 19th. And those dates are all going to be dependent on the weather. So if it looks like it's going to be pouring rain on one of those Sundays, then it's likely that it will be canceled. And another thing that will make a difference in those dates is if there's a need for mite strips or something that we can't open up, we can't open up the hives. So keep an eye out if you're interested. Anyone wishing to complete their practical test can show up at any of these events, and Ben typically at the end of each of these sessions will do the test. Or if you just want to come out to look to see what's going on, out at the hives, you're more than welcome. Proper safety attire is definitely recommended since you're working with bees that you're not familiar with. Okay, so I'm going to go ahead and turn it over to Dana. Dana is familiar to a lot of you. He's been helping us out with Bee School and some of our other projects that we have going on. And tonight, Dana's going to talk to you about when and why a beekeeper should replace his old comb. So I'm going to stop sharing and I'm going to go ahead and turn it over to Dana. And Dana, you're going to have to unmute.
Okay, I just
There you go.
unmuted myself. Yeah,
Okay, there you go.
I do appreciate seeing the former program. I was a former queen breeder myself, and somebody mentioned the fact that breeder queens are kept in smaller colonies, and that's very true. I've never really thought about size of eggs that queens produce, but that was quite informative to me. I'm going to spend a little time here. I've got a PowerPoint presentation. I'm going to share my screen and we'll get started on this and get through it as quick as I can. Do you see that? Hello? Do you see the screen?
No.
No.
Let me do this again.
No.
Share. I'm sharing my screen and hitting my PowerPoint.
You're not seeing it.
You're not seeing the PowerPoint at all.
So when you start sharing, select the PowerPoint, and then in the bottom right-hand corner, it should be share button.
Okay, let me, bottom right-hand corner.
After you select your PowerPoint that you want to share.
Mine is a green button.
I have no green button down here at all underneath my PowerPoint.
It should be on your Zoom window. At the very bottom middle, if you move your mouse up to it, there should be a share screen button.
Does that make any difference?
Not yet.
Maybe somebody can share their screen and show him what that button looks like.
Okay, I see share here.
There you go. We see your desktop now. Now we see your honeycomb management slide.
Okay, that's where we should be. I've been on this thing several times and somehow I'm still learning, okay? I was asked about, when should I phase out or replace old comb? And I thought honeycomb management is a pretty important topic. Most of you should realize comb is very important to the health of a hive of bees. And for new beekeepers, it shouldn't be too much of a problem because you're starting with new foundation. But for older beekeepers that have old comb in their hive, old comb can be a detriment. I just want to point out one fact, and that is in a standard deep hive, one side of the frame will produce 3,500 cells that a queen can lay an egg in. If she laid an egg in every cell on both sides of a frame, she would produce about 7,000 adult bees per frame. And when a person stops to think about that, that's 2 pounds of bees. Anytime there's any kind of deformity in a comb, that reduces the number of cells available for a queen to lay an egg in. And I just thought I'd point out there are 5 worker cells per inch in a honeycomb, and if you have drone comb, there's only 4 cells available. Why would you want to remove old comb from a hive of bees? Well, this is a journal, an article that was published that has done quite a bit of research on what they find in comb. In fact, I've read that there's over 50 different chemicals that have been found in old comb as they've examined what has come from the environment we live in. It includes pesticide residue and all of this exposure that's in that sink of comb in this old frame or this old comb has an effect on the reduced adult living age of a honeybee. I know the previous speaker spoke about 2 days longer with larger eggs, but a lot of the bee lifespan can be decreased considerably by the condition of the comb that the bee is raised in. Mortality is higher. Varroa mites seem to develop better in this old comb. There's a number of pathogens that could be passed on to the bees, so it affects the honeybee's health. And as far as resources needed to sustain a large colony of colony population, there are just going to be fewer cells available to raise an egg. Then this comes to a point I would like to get into: new beekeepers facing comb problems in new hives. Usually we don't think of too many problems that new beekeepers face, but there are a number of problems with the way bees will start growing comb, especially on plastic foundation. And anytime the comb in the brood chamber becomes disfigured, it could be burr comb or whatever, that disfigured area will no longer support worker bee cells, and it doesn't get any better. The bees don't correct that situation. So if you are a new beekeeper and you see bees building wax on the foundation, no matter where it's at, and it is taken away from the total number of cells that could be on that frame, it's a good idea to take your hive tool and scrape that comb down. If it's a plastic foundation, it's pretty easy to do. If it's not plastic foundation, it's a little more difficult to work with. Beeswax is a valuable product of the beehive, and if you save it, it's worth money. I have a solar wax melter, and I'm melting down burr comb and whatever I find in my hive all the time. Now, I'd like to have a few pictures here just to illustrate a frame that has bad comb and should be replaced. I don't care whether it's 5 years or whether it's 1 year. A comb like this is not going to contribute to the population growth of your hive. Instead of 3,500 eggs going into this side of this comb, the area that's available for the queen to lay eggs in is illustrated by the area within those blue arrows. And a lot of this, if I can point it out, this is drone brood down in here. You've got open areas in the comb. This comb could easily be recycled by melting it down. I'd like to point out the possibility also of using old comb like this in swarm traps. This is attractive to bees. Another issue that we run into is drone layers, whether it's a drone laying queen or drone laying worker. Comb is disfigured when drone larva is raised in worker cells. And this also is a sign that there's not going to be as many cells for a queen to lay worker brood in, and it will affect the colony size, how many new worker bees can be raised. And It also, if you take a look at these two frames I just showed you, this one and the one before, it also promotes swarming, because it's when you have a congested brood nest, bees will try to store honey here, but the queen has no place to put eggs. Swarming is really one of those events that happens to all bees, and it's triggered essentially by young bees not having any work to do, such as feeding the young larvae that should have been produced with eggs that hatched. This is a frame in the beginning stages of being disfigured. Notice that in the center of the picture, this area right here, there's a queen cell there. You have drone brood here. This is an example of a hive, maybe started this year, which had either a drone laying queen or laying workers. This comes from a student's hive that I have been working with, and came from a nuke rather than a package of bees. But the comb is fine up in here, but eventually this comb down here is going to be disfigured, and eventually over a course of years, it'll probably get worse. Here's another frame, same problem, a new package of bees in this case, bees trying to supersede the queen, and the queen is evidently a drone laying queen. She was in this hive, and this is on plastic foundation. And in this particular case, it wouldn't be necessary to remove the comb. We could easily take our hive tool and scrape off all of this comb. We could put this comb in a solar wax melter and recover it. You notice it's never going to get better. The bees may pull this area of the frame out into worker comb, but you're going to have a good portion of this frame that's going to be drone comb, and that will reduce the number of bees that could be raised on this particular frame. So I would take this out, scrape this wax off, probably recycle this frame by re-waxing it and putting it back in the hive for the bees to rebuild it. And I'm hurrying this along, by the way. Here's another frame. You've got broken bottom bars and things going on with frames. These frames need to be replaced. And I'd like to share with you, before I go back to the live screen, that in my bees, I take these old frames that I'm going to recycle, and I move them out against the sidewall of the hive so I don't get any brood in them, so that when I do replace them, I'm not dealing with trying to do anything to reduce my beehive population. So the bees that are here are all the worker comb on this particular frame has emerged. And at that time, the frame like you see here can be removed from the hive and put into a wax melter. There's a few cells down here that are drone cells. These may be worker cells here, but as soon as these cells are empty, it's time to take that frame out of the hive. And, you know, especially at this time of the year, while the bees are bringing in nectar sources, you can put a new frame in and they'll draw it out. And if they do it perfectly, you just increase the number of cells that that queen can lay in, and that results in better bee population in your brood nest. Now I'm going to.
Hey, Dana.
Yes.
This is Kim. Before you unshare your slides, we have Joe Tripp's asking on slide 9, which I guess is one slide back. Can you just remove the bad comb instead of scraping that entire frame?
Yes. This portion, can you see my arrow?
Yeah.
My cursor?
Yeah.
This portion hasn't been drawn out yet. One of the biggest problems with plastic foundation, I buy a lot of plastic foundation and I like to use it, but I get it double waxed. And it's very easy to scrape this with your hive tool, just like you're peeling the wax right off of the foundation. And then I like to go back and put another layer of wax on it, because I think anybody that's listening to this will share with you that if you take plastic foundation and re-wax it, the bees will really do a much better job of drawing the comb. For that person that's asking that question, I appreciate that. I would also say if you have comb that has a lot of pollen in it, I mean, I've seen total frames of pollen. Those are the kind of frames you've got to recycle out of your hive.
Okay.
But to me, a brood chamber should not have comb in it that looks like this, or this, or that. It just should not be found in the brood chamber, because it's promoting swarming. If you have swarms and you have old comb, you might have a better chance at doing swarm control with better comb.
Okay, sounds good. We're going to open it up for just a few minutes to questions. If anyone has questions for Dana, go ahead. You can unmute yourself and ask Dana your question.
This is Tamara here.
Yes,
I
Tamara.
know you said that now would be the time to take out bad comb, but is there a better time or a worse time to take out? I have a lot of black comb because I've been doing bees for about 10 years, and I'm trying to get it out, but you know, when you've got swarms and stuff coming in, you tend to want to put, you know, your drawn out comb in there, even if it is black. So
Right.
is it better to wait till later after the flow to try to do that,
Well,
or do that now?
Right. The bees are filling a lot of the comb up with honey. And I don't have any problem taking comb such as this is full of honey out of the hive. I do have a problem with brood on that comb. So if there's brood on it, I move it to the outside position in my bee box. I use 10 frames in my hives. So frame number 1 and frame number 10 would be on the outside. I always move my darker frames into those positions, and then normally I recycle my frames those 2 outside frames every year. So I'm taking 2 frames out and then inserting new frames in. And you know, right now the bees are really drawing really nice comb. And if I took this frame out, and by the way, I do have a frame that I took out of my hive today that I thought I could show you on my camera. But the thing is, take it out, it doesn't have any honey in it. It doesn't have any brood in it. And I can put a brand new frame in. You can checkerboard new comb between 2 combs that's already drawn out. And the bees will do a nice job drawing that comb out right now. And I would put it, if you have a double deep brood chamber, I'd start with the upper box and start replacing comb in that upper box, and then start working toward getting the bottom box. But it's going to take maybe 3-4 years for you to cycle all that old comb out, if that's what you've had over the last 10 years.
Okay, thank you. That helps a lot.
Yeah, it's really hard. It's really hard to, at this stage, I'm looking at my hives and they are wall-to-wall bees. They are bringing in a lot of honey. The queens are really producing a lot of brood. And if I run into a frame like this right now, it comes out. But I would not do that if the bees are actually utilizing, you know, the frame to raise brood. Honey storage, you could, if you're going to extract early, you could extract out of the brood chamber any of these old frames on the outside that are full of honey. And then I would suggest getting a solar wax melter. They're easy to build. Solar wax melter. I built one It didn't cost me anything to build it. Well, I went to the thrift store and bought a frame for $1, a big frame, you know, picture was in it. Took the picture out of the frame and just built a box around the frame and set it out in the sun, and it does a nice job of melting the wax.
We have a question here for you from Ken Cobb, Dana. It's what's the explanation for replacing combs of pollen?
If some bees are just really prone to collect a lot of pollen, and what every cell, the way I look at it, every cell that has something in it is a cell that a queen can't lay an egg in. And the key to my bees is bee population. So if I have a cell plugged up with pollen, I'll move it into one of my mating nukes or something. I've got, this year, I built 25 nukes, 5-frame nukes, and half of them are filled with bees, and I'm working on putting new comb in my hives, and I'm pulling frames out. My old frames sometimes go into these nukes to build them up. I've also taken my students that I'm mentoring, and several of them have swarm traps. I've donated old comb like this that went into their swarm traps. It's a good bait for honeybees looking for a new nest. So I know a lot of people use lures and things of that sort, but old comb is really attractive to swarms of bees.
Okay, and we have Jesse, you have your hand raised. Do you have a question? Go ahead and unmute yourself if you'd like.
I was curious to know, sorry. I was curious to know when you're using, when you're like trying to replace these combs, if you're feeding your bees, have you strayed away from 1-to-1 and tried to go even lower, like a 0.5-to-1? And how successful were you with that, with trying to get them to draw out these new frames that you put in the hive?
Okay, I started with this spring with 16 2-pound packages of bees put on drawn comb, and I started out feeding them, but they stopped taking the feed. There's so much nectar and stuff coming in that they're not even touching the food that I have in there for them in the feeders. And they are drawing comb like crazy. I could show you pictures I took today of brand new comb. It's just as pretty as you can see. And without any food in the feeders, because some of the feeders are empty. I always wonder about a hive that has food on it that they haven't taken it down. It's kind of a flag to see what's going on with that hive. Out of the 16, I only had 2 that had still some food on top of them. But I have deep hive bodies. I run my hives with a deep and then a medium. And the medium this year was all brand new comb. The deep hive bodies are now filled wall to wall with from a 2-pound package started in March, I think around the 20th. And they have drawn the comb mostly in the medium supers. And so I'm going to have to super them up with honey supers. Again, I bought 300 sheets of black plastic foundation for my medium supers. And I expect to have 30 supers drawn comb. I may have to, if this honey flow comes to an end, start feeding. But when I start feeding, it costs me probably $50 for each round of food that I buy for my bees. And I try to feed 2 parts water, 1 part sugar. Does that help you?
Yes, it does. Thank you so much.
Mm-hmm.
Okay, looks like we're getting to about getting close to 9:15. So any last comments or questions for Dana before we close up for the evening?
Well, Dana, I would just like to say I'm enjoying your emails that you're sending us with your thoughts and stuff for the beekeeping. I'm enjoying your notes.
Well, and let me tell you something. Those are like my lifeblood. It keeps me busy every week. I look at Saturday night as the night that those notes go out. Sometimes if something's going on here at the house and we've got company or something, they don't go out until Sunday morning. But I've already got this next week's notes done, and there's always a topic on beekeeping that I run across. My students provide me with ideas. Of course, I have a vast experience and a big collection of photo files, and every newsletter has something to do with this time of the season. You know, I've been asked whether I should archive these with the subject matter that's in them, but you'll find sometimes 2 or 3 subjects in a newsletter. And just like right now, this is the time that we have to deal with old comb. If you can take it out of a hive now and replace it with new foundation, you'll get your foundation drawn out. A little bit later this summer, it's going to be almost impossible to get them to draw out foundation unless you start feeding them with a lot of sugar water.
Hey, Dana,
Yes.
I have some folks asking, how can they get on your email list?
All they need to do is just say, add me to your list at AOL.com, which is Stallman Apiaries at AOL.com. That's STAHLMANAPIARIES at AOL. And it's free. I do it just for the fun of it.
Awesome. We appreciate it. Okay, if anyone didn't get that, we will make sure that in the yeah, there we go. Ken Cobb just put it in the chat, so we have it there.
And Kim, it's also in the newsletter.
Okay.
But
Okay. Well, thanks again, Dana. I know that you know, last meeting we had a little bit of a hiccup, and I think we resolved it. So thank you again for coming back and presenting for us, and we look forward to more of your mini sessions or big sessions, whichever.
Sure. And I'm always glad to help. I'm always glad to help.
Okay. All right. Well, I think we're going to sign off for tonight. Thanks again to everyone for joining us, and we look forward to seeing you next month. Our meeting in June will be June 8th, 7:30. And until then, be safe and good night to everyone.
Yep.
Good night,
Good night.
Kim. Thank you.
Good night,
Good work.
thanks.
Night, guys.